Okay. Good evening, everyone. We'll start today's live session. Let me share my slide and then we can start today's session. Okay. So let me share my screen. Okay. So good evening, everyone. Today's 7th live session of the series of live sessions for this course that we call RNA Biology. And in these live sessions, what we basically do is that we discuss some previous year assignment questions, and we not only discuss the question, we also discuss the concept behind these questions. So it's easier for us to solve similar types of other questions, right? So this is 7 live session of those trend of sessions, and we'll start with the first question for today. I'll show the question and I'll give you 4-5 minutes to think about it, and if you can answer it well and good, then we'll move on to the discussion part. So the first question for today is snRNA assembled site specifically on pre-RNAs. Is this true or false? I'll give you 4-5 minutes to think about the answer. Okay, so the answer will be true. Now, what are snRNAs? snRNAs are small nuclear RNAs. It's a class of RNA molecules that are found in the nucleus of eukaryotic cells. So, they are involved in RNA processing, including intron splicing and gene expression in regulation. So, the function is it's a part of spliceosome which removes intervening sequences from pre-mRNAs. snRNAs are involved in nuclear maturations of mRNA transcripts. They are also involved in gene expression regulation. How do they describe the structure? They are typically about 150 nucleotides long. snRNAs are transcribed by RNA polymerase II or RNA polymerase III. snRNAs are complex proteins to form ribonucleoprotein particles, snRNPs. And they recognize sequence elements in introns to form active lysosomes. Now, what are the other characteristics? snRNAs are extensively modified with different RNA modifications. They are said they bind to the sense and antisense DNA strand. They bind up and downstream to the transcription start site of their origin. Okay. If you ask for example, U6 snRNA contains sequences that are thought to be the functional counterparts of the RNA elements that form the active site of cell splicing 1 to intron ribozyme. And U7 snRNA is a small RNA with a big impact on gene therapy. Now, what are the discovery of first snRNA was found in late 1960s. Their band representing SN9 syndrome rat nuclei, high urine content led to naming convention. And there are several mechanisms of snRNA synthesis, biosynthesis. Each snRNAs are generally monocystronic. Plants generate polycystronic snRNAs, and vertebrate snRNAs are often within introns of our RNA processing genes. So there are several mechanisms of snRNA, as I said, polycystronic and intronic snRNAs require further processing to function. Okay, if you see over here, and snRNAs mediate the modification of other RNA nucleotides like these human 18S, 5.8S and 28S RNAs contain 110 2' O methyl groups and almost 100 pseudouridines. snRNAs direct methyl transferase and pseudouridines in this enzymes to the appropriate RNA nucleotide. And they act to guide nucleotide modifying enzymes to the proper RNA site. Antisense elements dictate whether the modifications are catalyzed. Okay. So basically, snRNA's function is a part of a ribonucleoprotein complex. If you can see the image over here. So what is the evolution? Enzymes likely evolved from gene duplications of tRNAs or RNA processing from enzymes. Ancestral snRNAs directly guided these enzymes and acquired specifications for novel rRNA sites. So they can be used as a biological tool as well because the localization elements have been utilized to direct new RNA sequences to the nucleus, and they have been used to direct nucleotide modifications to new sites in the rRNA via the expression of snRNAs containing novel guide sequences. So they have been implicated in numerous diseases like HB1/2/15 involved in modification of serotonin receptor RNA. Loss of either the parental or the maternal copy of this region leads to two neurological disorders, Prader-Willi or Engelmann syndrome respectively. Now what happens to the pre-RNA processing? There are terminal modifications like capping and poly-A-tail, capping at the 5' and poly-A-tail at 3', then there's splicing, intron removal, then there's editing, base chain, then chemical modifications, new mineral groups, pseudoiodine, HRRNA, methylation, and then cutting, pre-RNA 45S, miRNA biosynthesis, SNO-RNA biosynthesis. Genes exist in pieces, RNA splicing puts the pieces together. intron statistics. Nucleosomes are broken circles over here, preferentially bound to exons, whereas introns are mostly devoid of nucleosomes. Exons are therefore marked at the DNA level by nucleosome positioning, which may act as a speed bump for RNA polymerase II, helping in the co-translational recruitment of splicing factors to the nascent pre-mRNA and improving exon definition. As the nucleosomes accommodate DNA structures of approximately 147 nucleotide, their preferential location on exons may act as the selective pressure factor for the transition exon length. There are currently two opposing theories of intron origin. The introns early theory proposes that introns already existed in the progenitor, last common ancestor of prokaryotes and eukaryotes, to facilitate the construction of the first genes. The introns late theory, on the other hand, holds the genes of the progenitor were intronless, similar to those in the present-day prokaryotes, and introns were gained late after the emergence of eukaryotes. There has been no decisive evolution to the debate, and each of these theories has supporting arguments. What are the advantages of intron? Alternative splicing, create new exons, exon shuffling, create new exons, or intron exonization also. Then we have protein domains for exons. Many genes have evolved with exon duplication or shuffling. Similar exons are shared by different genes. Not going too much details. In not talk about this splicing. Two steps of amino I described this in the last class. If you can see the entire schematic of pre-amino. So the splicing proceeds in two steps. Number one, step 1, cleavage at the 5' splice site and location joining of the 5' end of the intron to the branch point A within the intron, producing a radial intermediate. Cleavage at the 3' splice site and the selection of the exons, resulting in inclusion of the intron as a radial structure. Number 2, transition reaction. The 5' phosphate of the intron is attacked by the 2' OH of the branch side and the side, causing cleavage of a 3 to 5' phosphodiester bond and formation of a 2 5 phosphodiester bond. Newly formed 3' OH of exon I attacks the 5' P of exon 2, causing cleavage of a new phosphodiester bond and formation of a new bond. The chemistry is simple, but the interval is in other words. Splicing, we are not going to splicing and spliceosome systems dynamic splattering in large complex consisting of 150 proteins, 5 small nuclear RNAs, and sample with proteins to form RNPs, alternative splicing factors. A spliceosome contains SNRNAs, SNRNPs, and many other proteins, typically about 300 subunits. This makes it the most complicated macromolecular machine known to date. Why spliceosomes are extremely complicated, different only catalyzes such a straightforward reaction as an intron to deletion. Even more, it seems that some of introns are separate to exercise themselves without aid of any protein. So why they have all those 300 subunits? No one's negotiable with reactive mRNAs cause a lot of problems for cells. So some units might assure cell slicing and error correction. Now, RNP consists of 10 different proteins and a nucleotide RNA molecule. 7 of the proteins, the SM proteins, are common to all U. It's an RNP. So, it's the other 3 are specific to the U1 is an RNP. Too much detail, you already know the structure of mRNAs. Base pairing between the pre-mRNA U1 siRNA and T2 siRNA early in the splicing stage, an experimental demonstration here that the first pairing between U and 5' bliss site in P1 is important. So splicing assembly pathway U for registration is U6 is an RNA to allow U6 is an RNA to pair with U2 an RNA to form the catalytic center for splicing. Both transition actions are taking place in the activated spliceosome. The splicing reaction is reversible at all stages. Steps in splicing, it ending of U1 and A2 and RNA base. Arrangement of base pairs, interaction between siRNA is realized of we not go into too much detail. Okay, not going into too much detail. I think for these slides. Order auto splicing. These splicing mechanisms in group 1 and group 2 cell splicing introns, spliceosome catalyzed splicing of PMRF. The discovery of cell splicing of ribosomal entry. Transplicing is different. The molecular splicing of PMRNA is South African trypanosomes. The mRNA is not encoded in the range gene called the spliced sequence. Spliced leader is encoded separately and there are about 20% of genome. And primary transcript contains 100 neutron that resolves the end of a 5 mRNA intron. The mechanisms of termination are different than in both in bacteria and eukaryote. In bacteria, the virus stops transcription at the end of the terminal and the membrane can be translated without further identification. In eukaryotes, RNA polymerase 2 shapes the polymorphism system sequence. The primer uses 30 minutes, 10 to 35 minutes in realize past this polymorphism sequence. So enzymes in the eukaryotic may modify mRNA before they generate genetic messages are expressed to the cytoplasm. During RNA processing, both ends of the primary transcript are usually altered. Also, usually some intermediate parts of the helicopter are cut out and the other parts spliced together. Each end of an mRNA molecule is played in a special way. Hypertrophy receives a modified nucleotide. 3' end gets a polyetail. They seem to facilitate the export of mRNA to cytoplasm. They protect mRNA from hydrolytic enzymes. They will determine attached to the 5' end. So most eukaryotic genes and the structures have long monopodial stages of nucleotides of tides that lie between growth and coding regions. These non-coding regions are called the intervening sequences or introns, and the other regions are called exons because they are eventually expressed, usually translated into amino acid sequences. RNA splicing uh, RNA removes intrusion, shines exons, and creating an RNA molecule with continuous coding sequence. Not going into too much detail. Okay, so we started the question. So we started the question with the thing. Key about snRNAs. We'll end the question now. We'll go to the next question for today. Length of poly tail on mRNA tail indicates the mRNA is undergoing decay, the mRNA is ready for translation, fusion of the mRNA and nuclear localization. I'll wait for 5 minutes. Okay. So the answer for this question will be age of the mRNA. So we'll discuss this. 1 second. Yes. So the last question answer would have been true, whatever we discussed. So now moving on to the next question for today. Length of poly A on mRNA indicates that mRNA is undergoing decay, that mRNA is ready for translation, age of the mRNA, nuclear location. We'll give you 2 more minutes. Okay, so the answer will be 8 of the mRNA. So what is this? Let's talk about synthesis of RNA transcript. The three stages of transcription: initiation, elongation, and termination. Right. So RNA polymerase binding and initiation of transcription. Promoter signal the transcriptional start point and usually extend several dozen nucleotide pairs upstream of the start point. Right. And transcription factors mediate the binding of RNA polymerase and the initiation of transcription. Now, the completed assembly of transcription factors and RNA polymerase II bound to a promoter is called transcription initiation complex. And a promoter called Tata box is crucial in forming the initiation complex in eukaryotes. If you see the schematic over here, there's a promoter, the Tata box, and you see the transcription factors are attaching. And then we have the transcription initiation complex. Okay. Now, what are the elongation of the RNA strand? As RNA polymerase moves along the DNA, it untwists the double helix 10 to 20 bases at a time. Transcription progresses at a rate of 20 nucleotides per second in eukaryotes. A gene can be transcribed simultaneously by several RNA polymerases. Nucleotides are added to the 3' end of the growing RNA molecule. Now, the mechanisms of termination are different in bacteria and eukaryotes. In bacteria, the polymerase stops transcription at the end of the terminator, and the mRNA can be translated without further modification. In eukaryotes, the RNA polymerase II transcribes the polyadenylation signal sequence. The RNA transcript is released 10 to 35 nucleotides past this polyadenylation sequence. Enzymes of the eukaryotic nucleus modify pre-mRNA RNA processing before the genetic messages are dispatched to the cytoplasm. Now, during RNA processing, both ends of the primary transcript are usually altered. So, also usually some interior parts of the molecule are cut out and the other parts placed here. What about the alteration of the mRNA ends? Each end of a pre-mRNA molecule is modified in a particular way. 5' end receives the modified nucleotide 5' cap and 3' end gets a poly A tail. These modifications share several functions. They seem to facilitate the export of mRNA to the cytoplasm. They protect mRNA from hydrolytic enzymes. They help ribosomes attach to the 5' end. This is the entire schematic: 5' cap, 5' UTR, start codon, stop codon, the 3' UTR and poly A tail. Now, most eukaryotic genes and the RNA transcripts have long non-coding stretches of nucleotides that lie between coding regions. These non-coding regions are called intervening sequences or introns. Other regions are called exons because they are eventually expressed, usually translated into amino acid sequences. RNA splicing removes introns and joins exons, creating an mRNA molecule with a continuous coding sequence. Pre-mRNA codon numbers 5' exon intron. If you can see 5' cap, 3' poly A tail. In some cases, RNA splicing is carried out by spliceosomes. Spliceosomes consist of a variety of proteins and several small nuclear ribonucleoproteins. It's an RNPs that recognize the splice sites. RNA transcript exon 1 intron, and you can see. Now, cell translates an mRNA message into protein with the help of transfer RNA. tRNAs transfer amino acids to the growing polypeptide in a ribosome, and translation is a complex process in terms of its biochemistry and mechanics. Now, molecules of tRNA are not identical. Each carries a specific amino acid on one end, each has an anticodon on the other end. The anticodon base pairs with a complementary codon on mRNA. If you see, it's 5' splice site, 3' splice site. If you see the 5' consensus sequence, 3' consensus sequence, and A is the branch point. This is splicing of nuclear introns requires a two-step process. The mRNA is cut at the 5' splice site. The 5' end of the intron attaches to the branch point. A cut is made at the 3' splice site. The intron is released as a lariat and the two exons are spliced together. The bond holding the lariat is broken and the linear intron is degraded. Spliced mRNA is exported to the cytoplasm and translated. RNA splicing takes place within the spliceosome. Spliceosome assembles sequentially. C1 attaches to the hyperlome splice site, U2 attaches to the branch point. A complex of U4, U5 and U6 joins spliceosome. U1 and U4 are released. Base pairing between sequences in the mRNA and the snRNAs hold the spliceosome together. Exons are joined together and the intron is released as a lariat. Intron is removed in the form of a lariat structure. Large intron is removed by splicing. With alternative splicing, pre-mRNA can be spliced in different ways to reduce different mRNAs. Either two introns are removed to yield one mRNA, two introns and exon 2 are removed to yield a different mRNA. So both alternative splicing and multiple 3' cleavage styles produce different mRNAs from a single mRNA. Now the gRNA are the guide RNA sequences that are partly complementary to those of the undeleted mRNA pairs with it. After this pairing, the mRNA undergoes cleavage and new nucleotides are added with sequences in the gRNA serving as template. Ends of the mRNA are then rejoined. Now the cap consists of 7-methyl-guanosine attached to the pre-mRNA by unique 5' bond. So one of the three phosphate groups at the 5' end of the mRNA is removed and the guanine nucleotide with this phosphate group is added. Methyl groups are added to position 7 on the base of the terminal guanine nucleotide and the 2' position of the sugar in the second and third nucleotide. The base of the initial nucleotide may also be methylated. So pre-mRNA is cleaved at a position from 11 to 30 nucleotides downstream of the consensus sequence, the 3' untranslated region. Addition of adenine nucleotides takes place at the 3' end of the pre-mRNA, generating the poly A chain. If we talk about protein synthesis, gene is a segment of DNA that codes for a protein, and gene's expression of protein synthesis is the process of using DNA's genetic code to make proteins. Transcription is RNA is written from the code of DNA in the nucleus, and translation amino acids are ordered in the proper sequence to form a nucleotide chain. In the cytosol of the cell, nucleic acid is converted to an amino acid sequence. Now, sugar is ribose, single-stranded RNA uracil, one is endocytosine bases. mRNA messenger RNA temporary copy of a gene that encodes a protein. tRNA is transfer RNA to globally shaped molecule that carries amino acid in translation. rRNA is ribosomal RNA with other proteins make up a ribosome formed in the nucleolus. SRNA is small nuclear RNA interact with specific proteins during RNA processing in eukaryotes. This is the tRNA structure, this is anticodon loop and the interpolypolar base pairing and etc. Now, sequence of bases of DNA or RNA that code for amino acid. Sequence codon 3 base pairs of mRNA that codes for amino acid. Anticodon 3 base sequence of DNA that is complementary to the mRNA codon. This sequence of amino acids. So, types of proteins made during synthesis are structural, antibodies, hormones, and enzymes. So, in the nucleus, DNA's code is transcribed into a molecule of RNA by transcription. Different types of enzyme RNA polymerase will transcribe mRNA, tRNA, and rRNA. rRNA and ribosomal proteins synthesized in the nucleus. A complementary strand of RNA is made from one strand of the DNA. RNA are modified in the nucleus that exit through pores in the nuclear membrane. Only one strand of the DNA, the coding of the template strand will code for RNA. RNA made during transcription will be complementary to the coding strand from DNA. RNA polymerase will attach to a section of DNA at the promoter site located just before the segment of DNA to be transcribed. Initiation factor proteins must be present for an RNA polymerase to attach to the promoter region. If we talk about elongation, RNA polymerase specially unwinds DNA exposing coding strand of the gene. RNA polymerase moves along the strand away from the promoter side as it adds complementary RNA to the nucleotides to make a primary transcript. So when RNA polymerase reaches terminal region, RNA polymerase and RNA primary transcript are released. So mRNA can last a few minutes to a few days, depending on the weights of process. Primary RNA transcript can contain up 20,000 nucleotides. Post-transcription modification can reduce mature mRNA by 1,000 nucleotides. All mRNA is processed before it leaves the nucleus. mRNA post-transcription modifications are enzymes and methyl group 1 starting tail of the mRNA GCAP. Enzymes replace part of the polyethyl oxidil with 100 to 100 adenyl nucleotides, polyethyl. MGCAP and polyethyl protect the mRNA from enzymes that would break down the nucleic acids. Polyethyl helps transport the mRNA out of the nucleus. Segments of mRNA that do not code for proteins are removed. Internal segment of mRNA that does not code for protein is segments of mRNA that code for proteins. Typically, remains after splicing. Removal of introns and designing of cut exons. The enzymes recognize G at one end of an intron and IG at the other end. tRNA processing. Nucleotides are modified and tRNA pellicle is folded into a stable clover-leaf shape with an anticoordinate coordination binding site. Primary RNA transferred to splice and modified into matured RNA molecules that will bind to proteins to form the two subunits of a ribosome. Now, the codon of mRNA is translated into amino acid sequence of a protein. Processed mRNA leaves the nucleus and enters cytoplasm and joins it to ribosome subunits. mRNA short codon signals a tRNA molecule carrying a methionine and attaches the anticodon at the next step. The next codon of the I side is the tRNA with the complementary exon carrying a specific amino acid. So enzymes in the ribosome form a peptide bond between amino acids, and the ribosome shifts down one codon. The tRNA of the B side enters the E side and introduces the methionine behind. The tRNA at the A side shifts to the P side, then A side is unoccupied. Only tRNA occupies the empty A side, the amino acids are bonded. This process continues until the stop codon reaches the A side. There is no tRNA with the anticodon for these codons. At releasing factor protein pairs with the stop codon and causes a new polypeptide chain to be released into the cell. The all schematics will not go. After translation, proteins need to be modified to gain a functional structure where proteins will fold to plain tertiary structure where sugars can be added to polypeptide like a protein. Proteins can be transported by air. And as a signal binds to an ES receptor during translation, and proteins is. Now, the starting point of a regulate reading frame can be off by 1 or 2 bases during translation. When mutation substitution of one base pair can cause a change in the one amino acid. Mature stop not to change at all. Frame shift mutation insertion of the base in the DNA sequence that will change every second codon. Tiny non-cellular particles that depend on the host cells for respiration, expression, and reproduction. Host cells, enzymes, and ribosomes replicate, transcribe, and translate the viral DNA or RNA to make new viruses which cause the cells to lyse. As usually, viral DNA is inserted in the host cell's DNA and is replicated when the cell divides. Bacteriophage to develop virus that attacks bacteria. Virus with RNA uses process of reverse translation to turn the bacteria into bacteria. Antivirus when cause viruses. You can viruses are used for vaccines. Reserve viruses can be used to obtain DNA for gene therapy and genetic engineering. Without too much details, the answer of this question would have been age of the mRNA. And anybody has doubt, they can ask me now, or we'll move on to the next question. Moving on to the next question for today. A disease that is the result of defective mRNA export is osteomalacia, osteoporosis, osteomyelitis, osteogenesis imperfecta. Anybody? So if you think about the molecular components of translation, a cell translates an mRNA message into protein with the help of transfer RNA, and tRNA transfer RNA to the growing polypeptide ribosome. So translation is a complex process in terms of its biochemistry and mechanics. The molecules of tRNA are not identical. Each carries a specific amino acid on one end, which has an anticodon on the other end, and the anticodon base pairs with the complementary codon on mRNA. So, the RNA splicing takes place within the spliceosome. Spliceosome assembles sequentially. U1 attaches to the 5' splice site. U2 attaches to the branch point. Complex of U4, U5, and U6 joins the spliceosome. U1 and U4 are released, and base pairing between the sequences of the mRNA and the mRNAs as an RNase holds spliceosome together. Exons are joined together and the intron is released as a lariat. Intron is removed in the form of a lariat structure. With alternative splicing, PMRNA can be spliced in different ways to produce different mRNAs. If you see the transcription, then PMRNA, 3' cleavage and polyadenylation. So, we have either two introns are removed to yield one mRNA, alternative RNA splicing, or two introns and exon 2 are removed to yield a different mRNA. So both RNA splicing and multiple 3' cleavage will produce different mRNAs from a single pre-mRNA. So with multiple 3' cleavage sites, there are two or more sequential potential sites for cleavage and polyadenylation. Use of different sites and produces different mRNA of different lengths. So the gRNA are sequences that are partly complementary to those of the unedited mRNA and pairs with it. After this pairing, the mRNA undergoes cleavage and new nucleotides are added with sequences in the gRNA serving as a template. Ends of the mRNA are then rejoined. So the cap, so one of the free 3-phosphate groups at the vibrant of the mRNA is removed, and guanine nucleotide with its phosphate group is added. Methyl groups are selected and added to the position 7 of the base of the terminal guanine nucleotide and the 2' position of the sugar in the second and third nucleotide. The base of the initial nucleotide may also be methylated. So the methyl cap consists of 7' 7-methyl guanosine attached to the 5' mRNA by a unique 5' bond. Pre-mRNA is cleaved at a position from 11 to 30 nucleotides downstream of the consistent sequence in the 3' untranslated region, and the addition of adenine nucleotides polyadenylation takes place at the 3' end of the pre-mRNA, generating the poly A tail. If you talk about protein synthesis, gene segment of DNA that codes for a protein. So gene expression protein synthesis is the process of using DNA genetic code to make proteins. Translation is RNA is written from the observed DNA in the nucleus, and translation and amino acids are ordered in the proper sequence from polypeptide chains in the cytoplasm of the cell. Nucleic acid is converted to an amino acid sequence. The central dogma replication, transmission, and translation. Messenger RNA is a messenger RNA temporary copy of a gene that encodes a protein. Transfer RNA for shaped biryani molecule that carries ribosome. Also under ribosomal RNA with other proteins become ribosome may be found in the nucleus. As an RNA, small nuclear RNAs interact with specific proteins while during RNA processing in eukaryotes. Sequences of bases of DNA RNA that code for amino acid, 3 base sequence of mRNA that codes for an amino acid, 3 base sequence of RNA that is complementary to a nucleotide, coded sequence of amino acid. Also proteins during protein synthesis, structural antibodies or northern enzymes. So in the nucleus, DNA code is transcribed into a molecule of RNA by transcription. Different types of the enzyme RNA polymerase will transcribe the RNA and transform ribosomal proteins in the nucleus. Currently, group of RNA is made from one strand of DNA. Data nucleopide in the nucleus and extrude in the nuclear membrane. Only one strand of DNA, the coding of the complete strand will code for RNA. RNA made during transcription will be complementary to the coding strand from DNA. So RNA polymerase will attach to the segment of DNA as a promoter right in 20 years before the bit of DNA to be transcribed. Initiation factor proteins must be either present for complex polymerase to attach the promoter region. So for elongation, RNA polymerase partially aligns DNA, exposing coding strand of the gene. RNA polymerase moves along the strand away from the promoter site as it adds complementary RNA nucleotides to break up primary transcript. When RNA polymerase reaches terminator region, RNA polymerase and RNA primary transcript are released. The mRNA can last a few minutes to a few days depending on the way it is processed. Primary mRNA transcript can contain 20,000 nucleotides. Post-transcription mutations can produce natural mRNA to 100,000 nucleotides. PolymRNA is processed before it leaves the nucleus. mRNA post-transcription modifications. Enzymes add a methylguanine nucleotide to the starting tail of the mRNA cap. Enzymes catalyze the middle part of the opposite tail with 100 to 100 nucleotides. Only here. Logic happen poly-A tail product mRNA from enzymes that would break down nucleic acid. The longer the poly-A tail, the longer the lifespan of the mRNA. Poly-A tail reduces the transport and out of the nucleus. The segments of DNA mRNA that do not code for proteins are removed. Internal segment of mRNA that codes does not code for protein Even segments a reminder that code for proteins remain after splicing. Splicing, removal of introns and exons. Splicing enzyme reconjugation choices, Q at one end of an intron and AD at the other end. tRNA processing. Nucleotides are modified and tRNA molecule is folded into a stable clover leaf strip with an anticodon and amino acid binding site. Primary RNA transcript is spliced and modified into mature RNA molecules that will bind to proteins to form the two subunits of a ribosome. The code of mRNA is translated into the amino acid sequence of a protein. M1 processed mRNA leaves the nucleus, enters the and goes into ribosome subunits. The mRNA start codon signals the RNA molecule carrying methionine and attaches to the anticodon same at the site. Next codon at the site receives a tRNA with a complementary sequence which is carrying a specific amino acid. Enhances the ribosome form a peptide bond between amino acids. The ribosome shifts down one codon. The tRNA at the P site enters the E site and that is itself the methionine behind. The tRNA at the A site shifts to the P site. The A site is now occupied. Unoccupied. The new tRNA occupies the empty A site. The amino acids are bonded. The process continues until the stop codon reaches the A site. There is no tRNA, the anticodon for these codons. A releasing factor protein pairs with the stop codon and causes a new polypeptide chain to be released into the cell. After translation, proteins need to be modified to gain a functional structure. Proteins will fold to obtain tertiary structure. The sugars can be added to polypeptide, glycoprotein. Proteins can be transported by ER. Amino acid signal sequence binds to the ER receptor during translation, allowing proteins in. After the signal sequence is removed and sugars are added, protein is transported to the plasma membrane or Golgi apparatus by vesicles. Starting points of a reading frame can be off by one or two bases during translation. Mutation changes in base, dew point mutation, omission of an oil base can cause a change in one amino acid, primary mutation subcodon, or in it all. Subtraction, mutation, and insertion or deletion of bases in the DNA sequence that will change every subsequent. So, length of poly A tail on mRNA tail indicates age of the mRNA. Okay, so this was the last question for today. If you guys have any questions, you can ask me now, or we can conclude the session for today. I'll stop sharing the screen. Okay, if nobody has any question, we can conclude today's session and we'll meet again next Monday, same time. Thank you. I'm sorry, the window got stuck and there were some network issues which I had to figure out. Yeah, sorry for the gap in the between. In the middle, I'll make sure that there is no network issues from next time. Thank you for the feedback. Thank you so much. Let's stop the session.